Welcome to our live session today. I will be going over a quick startup presentation to welcome you all and introduce you to our program and various opportunities if you have not previously attended our shadowing sessions. Let me just share my screen real quick. All right, can everyone see my screen? Can someone unmute and tell me?
Yes.
Okay. So I will be going over a quick startup presentation to welcome you all and introduce you to our program and the various opportunities if you have not yet previously attended our shadowing sessions. Pre-health shadowing is an international student-led, woman-led, minority-led nonprofit for helping prospective healthcare professionals gain access to educational resources no matter their demographic status, abilities, or location. My name is Tanusha and I'm a volunteer here at Pre-Health Shadowing. Thank you all for attending today and let's get started. Just a little PSA, we do have closed captioning. We do have closed captioning for all our sessions to accommodate all students, and the setting is available on the bottom of your Zoom screen. If you do need assistance enabling this transcript, please direct message one of our team members and we will help you. We are always looking for ways to be more inclusive and ensure our sessions are more accessible to everyone. So please, if you have any recommendations for how we can improve, you can email us at info@prehealthshadowing.com. So, since this is an international sort of organization, we do want to know, where are you zooming from today? Drop it in the chat. I'm calling in from Mississauga, Ontario, Canada. All right, that's really cool, everyone. So if you do want to stay in the loop and follow us on social media, we are active on Instagram and TikTok. You can also sign up for our email list on our website to never miss a session. We have some wonderful opportunities for you all as benefits of being part of our program. One of these is the research program. What this is basically a sort of research experience where you can connect with different PIs or principal investigators as they conduct research in a plethora of topics, fields, and locations. This program is 100% remote, and this means that you have the power to connect with anyone. This is an 8-week program that will begin on June the 21st. For more information, you can please fill out the interest form sent in the chat below, and we will get back to you as soon as possible with additional details. Furthermore, we have actually partnered with Kaplan. So with Kaplan, if you can get a 10% discount code that can be used on all Kaplan products, as well as the free resources such as study guides to help prepare you for standardized tests like the MCAT, NCLEX, or PCAT. If you fill out a short survey in the chat, we will get you signed up for these deals for free. We would also like to draw your attention to another amazing program, Neolith. Neolith is an online mental health platform for students and for pre-health professionals especially, because we know that the path is not easy. That's why we have partnered with Neolith to spread the word and offer free access to their services. If you use the link in the chat or enter the code pre-health when signing up. Additionally, we are excited to sort of draw your attention to our partnership with Krispy Kreme. Krispy Kreme, you can donate $10 to pre-health shadowing and get one delicious dozen donuts to enjoy with your loved ones. This will be possible by clicking the link sent in the chat right now. We also have the PHS pledge drive, which is where you can sort of get volunteer hours for every hour that you exercise. So to learn more information, you can go on to the link being sent in the chat right now. Another amazing sort of opportunity that we do want to bring you towards is Mask for Mask, which is an amazing women-led organization that donates 4 masks for every 4 masks sold. These go to people in need during the COVID-19 pandemic and those in the homeless community, healthcare workers with proper PPE too. The Mask for Masks also get donated to people struggling to stay safe during the pandemic going on right now. With our discount code PHS15, you can get 15% off your orders if you buy through this method, because pre-health shadowing will also get 10% of the proceeds, which is amazing because we are a nonprofit that runs solely on the support of a community. Yep. To play a bigger part in pre-health shadowing, we would love for you to join our network of student volunteers and team members, which you can apply to be part of our administrative team. We have lead students in various projects, and you can also interact with initiatives, professional outreach, grant writing, and more. We understand that as a pre-health student, you may not have the time, so we also offer the opportunity to volunteer asynchronously with tasks that can be done on your own time. We would love to have you be part of this program and contribute your own unique perspectives. If you are a high school student and want to get involved, we have started a program called HTP, or High School Training for PHS, which allows you to connect with college pre-health programs and get involved in fundraising for PHS and organize resources for other high school students interested in medicine through pre-health shadowing. We want to recognize. We also sort of want to recognize the effort that all our students put in the program. So if you are interested in getting published, you can submit essays, reflections, and research papers along with reviews to our editor-in-chief through the link dropped in the chat to have your work on our website. This will look great on CVs, applications, and resumes, etc. So take advantage of this. Part of our mission to promote diversity Part of our mission at PHS is to promote diversity. And in order to do this, we have launched an initiative to have monthly panels to celebrate different demographics in the field of medicine. Some of these upcoming events include a series on patent experiences, a COVID roundtable, and an international student forum. Through this, and if you have a mentor, professor, or professional that has inspired you and think you can contribute to these conversations, nominate them today using the link in the chat. If you can, we humbly ask you to donate to pre-health shadowing, because we are a completely student-run nonprofit organization, and we are working around the clock to keep this free and accessible to everyone. Unfortunately, Zoom and our website are not free, so any contribution you can give us would be greatly appreciated. If you're not financially able, we request you send this link to someone you think can, so we can continue to support those who cannot afford similar opportunities. Throughout the session today, we encourage you to drop any questions in the chat, or you can also unmute because our speaker will be having some time that would allow you to sort of ask questions. So we do ask you to ask questions to the speaker. We also will have some polling questions which you can interact with. Take good notes as our professional is going to go over the presentation. Lastly, if you can, we do request you turn on your cameras. This is by no means an obligation, as we are respectful of different circumstances, but it does help us feel closer together in a time when social distancing is mandatory. We also request that you make sure to mute yourself, as this will ensure the professional has the complete and full attention from the audience. I appreciate you for listening. Now I would like to welcome Ms. Garrett. Thank you so much for joining us today, and you may share your screen whenever you're ready.
All right, great. Do you guys see my slides?
Yes, ma'am.
Wonderful. Shall I start?
It's all yours.
Okay, great. So hi, everybody. My name is Francine Garrett-Bakelman, but feel free to call me Fran. It's really a pleasure to be here today and speak with you. And my goal today is to tell you about my academic journey and what I do today, ultimately, with the aim of allowing you to ask a lot of questions. And with that in mind, I encourage you to put questions into the chat whenever you think of them. And I've built in some points in my talk today where I'll just stop talking and we can have a discussion about what people are thinking and what questions you might have. So I'd like to make the session as interactive as possible, and that's one of the ways that I've found it to be fruitful from that perspective. So I was dually trained. After college, I enrolled into an MD PhD program. So I have an MD and a PhD, and I'll discuss that a little bit. And currently I'm an assistant professor at the University of Virginia. And I have appointments in the departments of medicine and in the department of biochemistry and molecular genetics. And I'm a member of the Cancer Center here at the University of Virginia. We are located in Charlottesville, Virginia. For those of you who may or may not have heard of this location, it's in the middle of Virginia, the state of Virginia, which is on the east coast of the United States. And I have no financial disclosures. So my journey started out in California. I had always wanted to be a clinician. That was kind of my childhood dream. And so I was a biochemistry major at California State University-Fullerton. And during my time there, I started to engage in research as a junior student, actually as a sophomore student. And I started out by working in a laboratory of Dr. Rogers, who was an organic chemist. And for me, that was really a disaster. I almost set the lab on fire a couple of times, and I just could not, it just wasn't working for me. I couldn't synthesize anything. It was just not my area of expertise. So in the last 2 years of college, I joined the lab of Dr. Robert Cook. He was a cell biologist, and I worked on him on cell signaling exploration in the creature called a sea squirt. For those of you who are not familiar with them, those are the little like goblets that live on the docks at the sea. I worked on how those particular animals facilitate fertilization. And that turned out to be a really great experience. Dr. Cook was great, great mentor, and somebody who taught me how to start to be a scientist. And it was through that experience that I became more interested in pursuing more scientific training, and that's how I wound up applying for MD, PhD training programs. So towards the end of college, I applied. I can't remember how many schools I applied to, but it was a lot. It's probably around 20. I interviewed at about 10, I think. And ultimately, I decided to go to Albert Einstein College of Medicine in New York City for many reasons. And this is something that, might be on people's minds. So if there are questions about how you make those types of decisions, once you have offers, I'm happy to engage in that conversation. So I went to Albert Einstein College of Medicine, and it was there that my professional direction was really set. So an MD PhD program, for those of you who are not familiar, is a dual degree program. And actually, we forgot to do something. Could we just do the polling questions real quick, just so I have a sense of who's attending? My apologies. So if everybody could enter answers to these as soon as they appear, that would give me a better idea of people's backgrounds and how to describe some of this information.
Awesome. So unfortunately, the polling system is down for today.
Okay.
But we can have the students put your answers in the chat for, you want to know where they are in their career journey?
Yeah, if some people could answer that, that would be great. Let me try and open up my chat function here. So if people could let me know, like, are you high school, college, first year college, second year college, gap years? Okay. Any postbacks? Wow. Okay. That's quick. Okay. Okay, great. That gives me some idea. Okay. And has anybody here heard of the profession of hematology? Okay. So it looks like at least most of the people who answered are saying yes. Okay. Has anybody here, do you know that you can get dual degree training with medicine, or is that news to you? Okay. Okay, so a mixture of that. What were my other questions? I can't remember. Can you guys help me out? I think there were one or two additional ones. I think one of them you guys already answered. I was just interested in knowing where you guys are calling in from. Was there another question that I had? Okay, great. Okay. Thank you, everybody, for answering that. Yeah, so when it comes to training and kind of graduate education, there are many options for dual degree training. Getting an MD and a PhD is just one option. With an MD, I can tell you that some of my classmates in my medical school class had law degrees, which they obtained before they came to medical school. And so that's another option that people do, is they do sequential training in different areas of expertise that they're interested in. But a lot of medical schools also offer dual degree training. MD, PhD is just one option. Other options include getting master's degrees, such as public health, master's in administration, and other aspects of career paths that can be pertinent to a medical career. So, if you have interests in more than just medicine and taking care of patients, maybe you're doing stuff now in your undergraduate or high school time that really fascinates you, and you really want to come up with a way to integrate that into your medical career plans, I would really encourage you to investigate and think about what dual degree programs exist. Most of them are integrated into your medical school education, so you don't apply to it up front. MD, PhD training programs are a bit different. You have to be accepted into both programs to be accepted into the MD, PhD program itself. But a lot of places, let's say you want to get a master's in public health, a master's in administration, maybe in mathematics or statistics to like enhance analyses, etc. Those are actually second degrees that you can explore after you join the program of the medical school curriculum. So just keep that in mind for those of you who might have more than one interest and really want to integrate some other expertise into your medical career long term. But for me, given my background of wanting to be a clinician, and then my scientific experience as an undergraduate, my path really was directed towards getting an MD, PhD training, and that's what I applied for. And as I mentioned, I applied to about 20. I think I interviewed at 10. And again, these interviews are combined. It's kind of a 2 or 3 day process at the beginning. You do the full interview day for medical school and then the full interview day for the PhD program. Both programs have to accept you in order for you to get an offer into the dual degree training program. So it was during that time that my career interests really solidified. I became very interested in internal medicine because I felt and thought that it was a direction in medicine that's very cerebral and incorporates really having to think about the entire human body on a daily basis. And within internal medicine, there are about 20 subspecialties. And at the time, my interest is really in immunology and how the immune system works. And that's what my PhD training was actually about. And so I thought that training in internal medicine would allow me access to a number of subspecialties that have insight and information contribution from the immune system itself. And also during that time, during my PhD, epigenetics was a new kid on the block. Does everybody know what epigenetics is? If I could see in the chat, just yes, no will be suffice. If I can open my chat. Okay, so a mixture. So everybody knows what DNA is, right? It's our genetic code. It's passed down from cell to cell, and passed down from our parents to us. So you can think of the genetics as being the hardware of your computer, okay? It's there, right? It exists, right? It can be replicated by a company who makes computers, right? Hopefully in a working order, not always, right? But in order to use the parts in your computer, right, you need software, right? Just like we're using Zoom right now to interact. So you can think of epigenetics as computer software, which allows you to utilize the code that's in our DNA to actually do anything in our cells, right? So when cells divide, right, the chromosomes condense, right, and nothing really happens. They just separate and you get two daughter cells. But then in order for those chromosomes to open up and allow gene transcription and allow proteins to be produced that could allow cells to do their job and to replicate again and do all these things, you have to have gene expression. So what regulates that are changes in the epigenetics of the chromosomes. And what those are non base pair changes or regulation of the DNA that are not changes in the DNA code. And there are hundreds of these things, okay? And it's the combination of these changes that determines what genes turn on and what genes turn off. And when do they turn on and when do they turn off? Meaning, when are they transcribed and when are they not transcribed? And why from one DNA component that each of us has, right, we can get all the organs in our body, right? All the genes are encoded in the entire genome. What dictates what genes are transcribed in the eyes versus those in our skin versus those in our heart, right? All of that is regulated by epigenetics, right? So my PhD work involved epigenetics. At the time, it was a relatively new concept. And it was there that I learned the fundamentals of what epigenetics is and what components regulate gene expression in cells. So, and so here is kind of a caricature of epigenetics. So here's your chromosomes and here's your nucleosomes. And you have your histone proteins represented by this red ball. And then you have the histone tails. And it's really these histone tails that get these covalent modifications done to them. But in addition to that, at the level of the base pairs, you can also get modifications to cytosines and other bases. So there's a number of locations where epigenetics can be effective and control gene regulation. And just an overview for the immune system, you know, in all of us, the immune system cells are produced originally in the bone marrow. And if you took a bone marrow from anybody who's healthy, it would look something like this, where there's this mixture of really heterogeneous cells, and each of them is a master of different aspects in our body. So this cell, for example, is a red cell, a young red cell, and that eventually will get into this person's blood and carry oxygen and carbon dioxide. These cells here are neutrophils, and these are cells really important in fighting infection. This cell here is a plasma cell, and those types of cells produce antibodies that help us also fight infection and respond to vaccinations, et cetera, et cetera, et cetera. And it's not this simple, because the bone marrow really originates from stem cells that produce a variety of progenitor cells. And through these progenitor cells, you can get a variety of cells, and all together, all these cells make up our immune system. And they're important for fighting infection. They're important for vaccination responses. They're important for the red cell production, as I described, and also for, of course, anemia conditions, right? And blood, regular red cell conditions that can happen. And then these small little guys here are platelets, and those are the guys that help us clot our blood when, like, we get an injury or something. Without them, you would just bleed to death, honestly. So this particular system is really life essential, just like the heart or the brain is life essential. And when it goes astray, people are extremely sick. So I then pursued internship and residency training in internal medicine. And it's there that I became very interested in hematological malignancies. So malignancies that come from the immune system. And I have to admit, when I entered training, I was going to be a rheumatologist because I was an athlete when I was young. And I felt this compelling need to take care of people like me who compete and get injuries, etc. And when you're in internship and residency, you do different rotations through the different subspecialties. And I rotated through rheumatology, and I hated it. And that was a total surprise to me. But that's part of the process, and that's why you have these rotations, to help you decide what to focus on. And surprisingly to me, and I didn't trust it initially, I really fell in love with hematology at that time. There was something about taking care of the patients that had this cancer type. You know, these patients are extremely ill. Many of them that I ultimately take care of today are in the hospital for long periods of time, and their life is really dependent on you and on the team that takes care of them. And so it's this very fragile time in people's lives where you can really make a big difference for people in a meaningful way. So what I ultimately focused on clinically and scientifically is studying this disease called acute myeloid leukemia, or AML. So what happens is, if you remember in my previous slides, this slide here, right, you start from these very immature cells, and from all these immature cells, you get all of these variety of cells. And so what happens in AML is these immature cells stop differentiating, right? They're like bad playing teenagers who just will not, they refuse to grow up, right? And so what happens in that situation now is you have accumulation of these premature or very immature cells called blasts. And these blasts take over the entire bone marrow space and prevent this normal process of hematopoiesis, or the development of the immune cells, from happening. And so then you get patients who can't fight infections because they don't have the infection fighting cells. They can't make red cells, so they become very anemic and have difficulty with breathing, etc. And they can't produce those platelets, and so they have bleeding complications. And so these patients, you know, if you don't diagnose them and start treating them quickly, they can die within a matter of days, if not weeks. So this type of leukemia is the most common one in adults. It is really a disease of aging. So most of the patients I see are in their 60s, 70s, and 80s. And in terms of, you know, who gets them, most of the time they're de novo, meaning there's no known cause for this. There are a few exceptions to this, such as ionizing radiation. I'm talking about really disaster level exposure, like the Chernobyl exposure or some disaster. Prior chemotherapy exposure from another cancer treatment, that's a known risk factor. Some chemical exposures, such as benzene and some other things. And then there are very rare genetic disorders, but in the vast majority of patients, we don't know why they got their disease. And in terms of how people do, you know, if this is the amount of disease that people have, they're diagnosed, and then you start treating them at this point. And thankfully, about 70% of patients respond to chemotherapy that we give them. But unfortunately, very few patients remain in what we call the remission state, which means that we think that we've gotten rid of the disease completely. What happens in most cases is that people develop the disease again, and that's called relapse. And then there's a smaller fraction of patients that are refractory to treatment altogether, and we can never really get rid of their disease. And unfortunately, the trend for this disease has not really improved. So this is from SEER data, so this is US data, but if you look worldwide, it's very similar. And you see that between 1975 and 2016, there's a steady rise in cases that are happening, probably because the population is aging and because more and more people are surviving chemotherapy treatments otherwise, so they're at a higher risk. And unfortunately, the death curve of these patients is not changing either. So even though there's new therapies and we've made advances, etc., most people will die from this disease if they're diagnosed with this disease. So let me stop here for a couple of minutes and see if people have any questions about kind of career path and or about acute myeloid leukemia. And if the moderators could help me kind of understand if there are any questions to look at. Okay, I see them. Okay, so the first question is from Alexia. Are stem cells just all the cells in our body? No. So there are stem cells in organs in which there's regeneration of cells. So for example, in our tongue, right? Remember, like when you burn your tongue, right, you lose sensation. but then over time you regain it again. Those are stem cells that are starting to generate new cells again. The same process happens throughout the gut, actually. You shed a lot of cells in your gut, and those are replaced by replication and differentiation of stem cells. There are actually stem cells really in all of our organs. It's just that in some organs more than others, they replicate and produce new cells on a regular basis. Before they are determined what they, okay, all right. How much research experience do you need in a wet lab to be competitive for an MSTP? MSTP is an MD PhD program, and does translational research count as well? So I think, you know, how much is really dictated by the school that you apply to. The applicants that I review here at UVA and people that I looked at Cornell, which is where I did my first faculty position, on average had between 2 to 3 years of research experience, but I've seen people come in with less. And some people also do like a post-bac year or gap year in which they do full-time research, which of course is much more productive from a research perspective, as opposed to doing 10 to 20 hours a week as a student, which is what I did for 3 years. And that's really just my perspective. I think if you have very specific questions about specific schools, you should direct them to those particular programs. And does translational research count? I personally think it does, but I think it depends on the school that you're applying to. So if the MD PhD program that you're applying to is very supportive and very heavy on the translational aspect, then I think it's totally a fair game. But some are not. Some are much more hardcore basic science type of places. So again, this is something that I think you should pose as questions to program directors or administrators, and you should not hesitate to reach out to them early in your thought process, because clearly it takes time to gain these types of experiences, and they should be very transparent about what they expect in their application pools. Is there any knowledge as to why there is a high rate of relapse? I'm assuming that's about acute myeloid leukemia. You know, that's an active area of research. That's something actually that my lab works on actively as well. You know, the trains of thought is that either we don't do a good job treating patients up front, and we just never get rid of the disease, even though we think we did. And so the cancer cells are still there, and as soon as the chemotherapy is gone, it just lets go and returns. Other trains of thought is that exposure to treatment actually stimulates new cell types to develop that can have cancer potential. And so that's another train of thought. The third reason is that as people age and as they get exposure to toxic chemicals such as chemotherapy, they gain more and more mutations in their genetic information, and through that they have a higher risk for disease. So those are some of the reasons why we think people relapse so highly in this disease. Yeah, so there are some questions about specific treatments. You know, a lot of these newer treatments are experimental and not yet tested fully in what we call randomized clinical trials against the standard of care. So it's hard for me to comment on those right now. But if you have specific questions, and this is from Alexia again, there are clinical trials that are listed on like the National Cancer Institute websites that you can go to and follow them along. And you can feel free to reach out to me after this session if you have more specific questions to address. What makes AML resistant to its therapies? Yeah, that's a great question. So again, there are quite a few theories. Some of them, if you treat patients with targeted therapy, there's some research to suggest that specific mutations or other events in the cells are gained that cause very specific resistance to specific treatments. But when people are resistant to the more standard chemotherapy that's not necessarily targeted, we don't really fully understand why they're so resistant. In some cases, there are patients that have expression of proteins that can either pump chemotherapy out of their cells or break down the chemotherapy quicker than in other people. So the effect of the chemotherapy is reduced. And in some people, we just have no idea. And unfortunately, we also don't have a good way to predict. So you don't know who these patients will be most of the time until you start the treatment. Yeah. What is the most rewarding / fulfilling aspect of my career? So let me save that question until I get a little bit more into my presentation. I'll just check the time here. Okay. How much of your time is dedicated to research versus clinical work? I'll go over that in a couple of slides. Is there a common age of diagnosis for individuals with AML? So the average age of diagnosis is 68, and most patients are over the age of 60. So, most of the time when you think of AML, you always picture the children that are, poster childs on TV and ads, right? But actually, the young children are the minority of the patients that are treated, and the vast majority of people are over the age of 50, with the most patients usually in their 60s and 70s. What is the difference between MD PhD program compared to getting a PhD and then deciding to go to an MD program after? So the difference would be that you would have to apply to the PhD program, and then whenever your PhD is done, apply to the MD program separately, as opposed to doing them combined. In theory, the combined program takes less time, in theory. It depends what the course requirements are at the school. But a lot of times there's a lot of overlap between medical school and graduate school, depending on your area of expertise. So some of the courses count for each other, right? So you can kind of save some time. You can shave some time off that. The other advantage for those of you who this is important for is that, when you go to medical school, unless you have a scholarship, right, the tuition is billed to you in full. MD PhD programs traditionally at most institutions that have these types of programs that are supported by the National Institutes of Health, they basically are sponsored by the institution. So you might have some debt for other things, etc., but your tuition is covered and you have a basic stipend that is paid. So that's a big difference in that respect as well. Okay, so I don't see any other questions right now, so let me move along. So I did my internship and residency at Weill Cornell Medicine in New York City. And then I actually had a gap year because of that decision where I didn't realize I wasn't going to like rheumatology. So my decision for a subspecialty application actually was deferred by a year. So I had a gap year. And so what I did during that time is I did a very short postdoctoral training. So for those of you who are interested in this kind of stuff, even when you get your medical school degree and even when you get your scientific degree, you still have to do postgraduate training in order to really be eligible to work full time. So this was my first postdoctoral training. I did it with somebody who was on my thesis committee at Cornell. And then I had decided to apply for the hematology oncology training. And I stayed at Cornell as well. And then after that, I joined the faculty as a junior faculty member at Cornell. I joined as an instructor, which means a lot of things at different institutions. For what it meant to me it was my second, or I consider it my real postdoctoral training in the laboratory. So I spent 85% of my time in the lab doing research with my PI. But I was also a practicing physician because I had completed all my clinical training. So I was taking care of patients in the hospitals and attending. And I was in that position for 4 years. 4 years plus, I can't remember anymore. And then in 2017, I moved here, I was promoted for about 6 months to an assistant professor at Cornell. And then after that, I moved to Virginia, which is where I am now. So during my postdoctoral times and during my fellowship training and during my instructor position, I worked on research throughout. And, you know, AML is a rare disease, so you can't study it in isolation and alone. And so through connections that my PI had and support from him, I really became familiar and worked with a variety of people from all over the world. And that was just really a great experience for me. And these people that I met and worked with during that time continue to be my collaborators today, where I have an independent position. And so I'm very thankful for that experience at that time. And so after I did that short stay as an assistant professor at Cornell, I moved to the University of Virginia. And here I'm a physician scientist. I have a basic research laboratory that focuses on the study of AML and the pathogenesis of AML, so why people get it. And I focus on high-risk populations, so people who relapse and people who are over the age of 60. And both of those populations have very poor clinical outcomes, unfortunately, and we understand the least about their disease. And so that's what my lab focuses on. My lab is a combination of bench scientists as well as computational scientists, because in our profession, there is a lot of genomics data. And it's really part of the course now to understand biology through analyzing genomics data. And so that's really a necessary component of any lab in our field. Now, you know, I didn't get here by mistake necessarily. There were people who really motivated me along the way, and I just wanted to highlight a couple of them. And so Dr. Cook, I mentioned, he was really my first true PI in the cell biology department at Cal State University-Fullerton. And then my MD PhD program director was Betty Diamond, who's also a physician scientist, and she was really an awesome role model for me in helping me direct my career forward. Barbara Burstein was my PhD PI. She was a cell biologist, and she had never done epigenetics before I joined her lab. And so her and I together embarked on this crazy project that became my thesis PhD project. And it was really a lot of fun. And I really learned a lot from her and credit her for my scientific maturation, really. And then Ari Melnick, even though he's an MD, he does not practice medicine. After his clinical training, he did a postdoc and decided that he just wanted to be a scientist. So for those of you who don't necessarily want to commit to a dual degree program, you know, the other options include doing an MD and then doing a prolonged postdoctoral training period to give you the training and expertise of a PhD level person. And he is really now a world leader in the study of hematological malignancies. And nobody cares that he doesn't have a PhD. Let me tell you that. Okay. So he's just doing great. And his career is really flourishing now. And so Ari was my postdoc PI. He was also on my thesis committee, my PhD thesis committee at Einstein. But he moved to Cornell soon after I started my training there. And so I worked with him on epigenetics and continued the work on understanding AML biology with him. And then the other thing I want to highlight is that along the way, there are many training opportunities that people can take advantage of. And so I just wanted to highlight a couple here. So in hematology specific, there are two main organizations that really are major components of where people get information, research is done, etc. And this is the American Society of Hematology and the European Hematology Association. And together they have some training programs for junior faculty. And I participated in one that was focused on research called the Translational Research Training in Hematology. 20 trainees, 20 faculty, so it's a 1-to-1 ratio. And you really, they really help you with an idea for a project. And actually, what I had worked on during this time became one of the main projects that I work on in my lab today. So I'm very thankful for that. And then I participated in a couple of leadership and laboratory management training programs, one at Cornell when I was faculty there, and one that's run by a nonprofit organization called EMBO, which was in Europe, and I traveled there to participate in their program. And since then, I continue to participate in training programs to enhance my ability to mentor people who come through my lab on a regular basis as well. So, so overall, this is where I am now. I take care of patients that have hematological malignancies. I study AML. And with the idea that the disease mechanisms in these high-risk populations are not really understood, and if we can understand them, maybe we can help identify new clinical treatments that can improve clinical outcomes. So I want to go back to two questions that were here. How much of your time is dedicated to research versus clinical work? So the way I divide my time is, and this has been the case since I was an instructor at Cornell as well, I spend 1 month a year attending in the hospital and helping to take care of patients that have blood cancers. And that's divided into 4 individual weeks. So usually once every 3 months, I'm in the hospital working for a week, 7 days straight. And then the rest of the time, I'm in the lab and taking care of business there, right? Writing papers, grants, supervising students, postdocs, undergraduates, helping with work in the lab, etc. So that and that works for me really well. It's not for everybody, but it works for me very well. And then the other question was, what is the most rewarding, fulfilling aspect of your career? So I would say at this point, there's really 2. The first is the privilege and the ability to help patients that have this type of cancer and help their families get through a very difficult time. Unfortunately, in terms of patients over the age of 60, which is the ones that I see the most, their outcome is not great. Their median survival is 1 year. So, if you can really help them get to that 1 year mark, or maybe less, maybe more, but you allow them to attend a graduation or see their grandchild being born or whatever it is that their goal is, it's really a privilege to be able to do that and help in that process. And the other aspect is helping people in the lab get trained and mentor students and trainees that come through. And at this point, even though I'm new to this as a PI, I already have people who came through as employees and now they are graduate students themselves. And it's really fulfilling to see that development in people over time. The research that I do is focused on AML biology. We are publishing information about this. It's very fulfilling to be able to contribute to the literature as well. But, as a physician scientist, your ultimate goal is to contribute to a therapy development. I am far from that. Usually it takes 20 to 30 years to accomplish something like that. I hope I reach that. I don't know that I will, but just going through the process and contributing to the literature that hopefully will work towards that goal overall is very fulfilling. And that's what keeps me motivated along the way. So let me stop here and see if there's any questions at this point. And if the moderators could just let me know how much more time I have.
You have around 20 minutes left.
Okay.
Yep. And
Okay, thanks.
one of the questions that is in the chat is, what advice do you have for undergrad students trying to get research opportunities?
Yeah. So right now it's very challenging. I think as vaccination rates increase, opportunities will become more and more available. But I think it's going to be hard, especially if you're in a country where the vaccination rates are not that great right now, meaning less than 50%, which is really most of the world at this point. So I would say, I think you want to start early. You really want to start as a second year, if not before. And what you're looking for is a mentor who will help you grow and learn, right? Whether you get a publication or not, in my opinion, is not as important as what you learn along the way and the scientific understanding that you gain through that experience. So I think, looking for a lab or a PI, if it's like clinical translational type of research, it will give you an opportunity to learn something new or utilize skills that you have to apply it to real world data, is a great place to go. I think you have to be careful about who you join and talk to people in those labs and find out, what their mentoring style is, what people's experiences have been, good, bad, indifferent. And you really want to try and find a place where you can envision yourself being productive and learning, and not just join any random person or lab to do a project, because that will really make or break your experience. And keep in mind, you probably want that person to write a letter for you when you apply. So this is why that type of matching process is really essential. So that 2 years later or whatever, 6 months later or whatever it might be, you're in a position that you can, get some work done and really be in a position to get a good letter of recommendation from that person.
Mm-hmm. And someone asked Rosemary asked in the chat, what is your advice for making the most of research opportunities?
Yeah, I think that's different for every person. I think being paired with somebody in the lab who's working on the project that you're working on is really helpful, because you can be engaged and active all the time, not just when the PI has an hour to meet with you. I think really being proactive about the work and, not just showing up and doing work that's assigned to you, but actually thinking about the projects that you're involved with, maybe exploring some literature out there that might be pertinent and bringing it back for discussion, thinking about experiments or analysis approaches, if that's what you're doing, and coming back with ideas and recommendations that people could think about and incorporate is really important to consider.
Mm-hmm. And you mentioned that AML, it's a really fatal disease and you can get really hurt, you can get attached to patients. What is the emotional toll of working with such a fatal disease? And what if you're interested in going into something like oncology, but you're very sensitive to dealing with harsh treatments and death a lot? What would you recommend?
Yeah. So that's a very tough question. I think different people deal with it differently. But if you go into oncology or hematological malignancies, it's part of your career. And so it's something that you have to be prepared for. So for me, the way I focus on it is I try and make those last days or last weeks or last months as good as possible for people. And I take fulfillment in that, maybe I relieve some of their suffering, right? Even though we know that the end game is that they're going to pass away before they wanted to, or anybody wanted them to. And so it's really hard. There are days that I cry with patients and their families because we're human too, right? And there are days when, somebody passes away and they're in a better place because they were suffering at the end. I talk to my colleagues, I talk to my friends. I usually do not bring it home. I do not talk about these things with my family. That's kind of a principle for me. I keep work as much as I can out of the home, or at least out of business hours from home, since I'm at home right now doing this. But it's really important to, be able to do that. And different, a lot of institutions have programs set up to help clinicians deal with these stressful situations. I personally have not taken advantage of those programs, but those programs are available because institutions realize that this is a really stressful job. Even not oncology, right? I mean, you go into cardiology, you're going to see the same kind of stuff, right? You go into kidney failure or whatever, you're going to see the same kinds of stuff. It may not be as frequent. It may not be happening to as young patients as we see, right? But that's part of medicine. But this is part of the selection process. Like if you're not comfortable with being there to help people through this type of process, then maybe another subspecialty might be better suited. And this is where those rotations come in when you do in medical school and then in residency. And so you have multiple opportunities to really explore these feelings and these thoughts and really come up with an educated decision about your career direction. But it is hard. I'm not going to lie about that.
All right. Thank you. I think there's one last question in the chat. How do you deliver news to families that may involve the death of a loved one?
Yeah, that's, it depends on the family. That's a very complex question. I'm not sure I can really give an adequate answer in the short time that we have. There are teams of physicians that really work together to care for these patients towards the end. And to me, the most important thing is that people are transparent about what's happening so that if death happens, it's not a total surprise. People deserve the truth. And even though we sometimes think that it's not the right thing to do, or they may not want to hear it, most people want to hear it. And that also helps people prepare, right? Because then they have the time to do what they need to do in preparation for that. And I think that's really important. But, the words that you choose and how you do it if there's young kids involved, et cetera, that's an art that you have to learn. And that takes time. I can't really explain it in a couple minutes. But if you're going into this field, it's something that you will learn and you will get exposure to, and you'll figure out your own way of how to do it in a very sensitive way.
All right, thank you. That's about it for now.
Okay. So just a couple more slides. In research, you have to have funding, otherwise the lab closes. So these are my funding sources and some of the funding sources that helped support the work that I did as a postdoctoral, as the instructor position and postdoctoral fellow at Cornell. In my laboratory, what we study, as I mentioned, is the disease pathogenesis or the disease biology of AML. And this is just to give some overview of how we do this. So we work, as I mentioned, we have what we call wet and dry bench scientists. So wet bench is at like the actual bench, we're using pipettes and whatever. And dry bench, we call people who do computational biology, so the analysis part. And so what we usually do is we start out with what we call cell lines, which are cells that at some point were isolated from an AML patient, and they naturally continuously grow in the laboratory. So they're in some way transformed that allows them to survive in the laboratory and grow indefinitely, or clinical samples from patients. And most of the studies in my lab, we are asking questions about specific genes and if they are causative of the disease. So we use things such as CRISPR-Cas or chemical inhibitors to perturb those specific genes and understand what their role is in the cell biology. And once we have these treated cells, we can do a lot of things. We can do genetic analyses and epigenetic analyses that I described earlier, and also gene expression to try and understand how these genes work and what biological processes are controlled by these genes that might be susceptible to therapeutic interventions. We can grow them in culture in the laboratory, both in a liquid form and in a semi-solid form. And through that, we can assess what aspects of cell biology are affected. Are the cells proliferating or not proliferating? Are they dying? Are they differentiating? Like, what's happening to them when you perturb these genes? We also do, you may have heard about single cell genomics assays. So this has become more and more dominant in our field. It's been recognized that AML is a very heterogeneous disease. So in one patient, you might have a number of different clones of cells that have different genetic backgrounds. And so in order to appreciate the biology fully, you actually have to study it one cell at a time and understand what the gene expression is there and what molecular events happen there. So in order to do that, you can do single cell analyses. And then ultimately, once you identify some biology or some gene or some pathway that you think is really important in the laboratory, you have to test it in some model. So most of the time in our field, we use mice that are AML models. Some of them are genetic models, meaning that the mouse itself develops disease spontaneously. But many times what we do is we use mice that are essentially treated with chemotherapy or some radiation to get rid of their bone marrow cells. And then we transplant them with a human specimen of AML. And then we can do experiments with chemotherapy or perturbing particular genes to see how these cells survive or don't survive, whether they kill the mouse earlier than what you would expect, or maybe the mouse survives longer. So these are kind of the types of assays that we do, and we use all of this to really understand disease biology. And the theories that we're trying to answer are, when you have a patient that's diagnosed with AML, what are the genetic mutations that might be contributing to the disease and how are they contributing to disease? And on the other hand, as we discussed earlier, what are the epigenetic characteristics of that disease that might be allowing these cells to survive long-term and really cause havoc in patients? And that would include gene expression regulation and the epigenetic modifications, et cetera. And to me, it's really important to study that because epigenetic changes are potentially reversible, as opposed to these genetic mutations that are permanent. So if you can identify an epigenetic target, you could potentially come up with a therapeutic modality that could reverse the effect and cause those AML cells to basically die or become mature and normal and function and basically potentially cure the disease if that is the dominant or the only event that's happening in that particular patient. And so we study these types of changes, and then we also study what these drivers are contributing to the relapsed disease. So my lab focuses on questions relating to how disease biology works in relapse and how disease biology works at diagnosis in patients over the age of 60 as representatives of high-risk patient populations. So that's my last slide, and I want to thank everybody for your attention. I hope this was educational, interactive, and helpful to people, and I'm happy to answer a couple last questions that people might have.
So there's one question on whether one should take a gap year or not. I know that you mentioned you taking a gap year. What do you think are the sort of indicators that someone might want to take a gap year?
Yeah, I think it's a very personal decision. Sometimes, there's something personal, family, or otherwise that would prevent you from going straight through. And that's totally valid. Sometimes people need a break from studying all the time. Once, grad school, undergrad is hard, grad school is harder, and med school is harder, I have to say. And then internship is harder. And residency is harder, and fellowship is harder than that. And then once you get to your position, it's harder. So, this is this kind of lifelong continuum, right? And what I tell people is this is not a sprint. This is not about who gets to the finish line first. This is a marathon, right? And the point is to get to the end of the marathon. So if you need a break from this crazy studying, testing, blah, 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 whatever, take it, right? And do what you need to do to help yourself get to that finish line. Some people feel like they need more research experience, so they use that year for that. Some people feel like they need to improve their GPA, so they do a post-bac training, right, to help improve their GPA. Some people maybe had a bachelor in arts, right? And then they decided, oh, I really want to be a physician. Well, then you need to take all the pre-health stuff, right? So then they go back to school to take all the pre-health requirements. I think it's a very individualized situation, and I would encourage you to talk to your advisors about what's best for you.
All right, and we have one more question about when new undergraduates enter a research laboratory, typically how long does the training take?
Yeah, I think it depends on the lab. So I can tell you what I do, okay? So in my lab, the way we work with undergraduates is for the first semester that they join as a volunteer. So I don't expect them to be in the lab a certain number of hours. And it's really up to them how motivated they are and proactive they are about it. I recommend being in the lab at least 10 hours a week because it's really hard to accomplish anything in less than 10 hours a week. And then we use that time to kind of mutually evaluate things, because sometimes you join a lab and you hate it, right? It's just not for you. So, no hard feelings, right? At the end of that semester, we're done, right? Period. And on the other hand, if people really like it, it also gives the lab an opportunity to interact with you and see what your skills and capacities are. Because, if it depends on the lab, like my lab is very small, so we don't have a lot of people and manpower to train. So the people who are accepted into my lab for like class credits, etc., are people who've been in the volunteer position for a semester and who have really shown promise in learning how to implement things, how to do analyses, how to do experiments. And we feel like they can fit really well into the workflow of the lab and contribute to projects that are ongoing. But every lab is different. And so I encourage you, if you're looking for these types of experiences, talk to the PI in a very transparent way about what the expectations are and kind of how that develops in each of the labs, because every lab will be unique. The only thing I will say is, avoid labs where you might just be cleaning glassware or, finding just, copying papers or whatever. That is not a productive use of your time. That is part of being in a lab. So yes, you would expect to do that to some level, but it should not be 100% of your time. And you should be engaged in some project at some level so that 2 to 3 months later, 6 months later, a year later, you can actually talk about a scientific project that you contributed to.
All right, thank you. There's just 2 last questions, mainly pertaining to high schoolers, because we do have a lot of high school students in the watching right now.
Yeah.
Do you work with high school students in your lab, and what is a small piece of advice, like a 1-sentence piece of advice you can give to high schoolers right now?
Yeah, so currently I do not have any high school students in my lab. My lab, again, is new and small. And so I'm not quite at the point where we're developed enough to give high schoolers a really good experience yet. But I'll probably do that in the future. What I would encourage you to do is a lot of academic institutions have research experiences, especially in the summer for high schoolers. And so I would advocate that you research those and apply to them and participate in those, because they really expose you to research in general, and they can match you with labs in which your exposure and experience will be really optimized in an 8-week period. But I have to say, I do know some people who did do research during high school, and independently they found PIs and labs to work with. And so, I think you have to be very motivated and proactive about it. But I would first find schools that have programs geared towards this, because more than likely they will have a lot of PIs who can accommodate high school students in their labs at any time.
All right, thank you. That's about all the time we have right now. I do have a wrap-up presentation that I need to go through, so can everyone please stay in the Zoom meeting? We are going to be giving you lots of information. There are other questions in the chat, but we simply do not have the time as we are running short on it.
So, if I could recommend, if you guys want to collate those questions, I can take a look at them, answer them, and then you can circulate it to the attendees if that's possible. Is that possible?
I'm not entirely sure.
Yes. So we have a transcript of the chat that we can send the questions via. So thank you so much for doing that.
Yeah, sure. Happy to do it.
All right.
Thank you again for the opportunity.
I'll start sharing my screen now for the, yep. This is the wrap-up presentation. Thank you so much for that informative presentation, Dr. Francine Garrett-Bakelman. I think we all learned a lot from our wonderful speaker. And considering this, we would like to encourage everyone to reflect on the session today. What brought you to the session today? What are the 3 major takeaways that you got? What do you want to learn more about? This writing or reflection is not required. However, we do encourage you submit it to our website for publication and for recognition of your hard work to enhance your future applications. We sort of did touch on it in the starter presentation. I would like to mention once again that pre-health shadowing is having a research program, and that will allow students to connect with PIs or principal investigators. This is not available to high schoolers, but this is available to undergraduates. And as they conduct research and these researchers conduct research in a plethora of topics, fields, and locations. This program is remote, which means you have the power to connect with anyone. This is an 8-week program, and it will begin on June the 21st, which is kind of past, but you can reach out to us for more information. You can also fill out the interest form sent in the chat below, and we will get back to you as soon as possible. If you want to learn more about pre-health shadowing and get involved in our in our program, you can do that. You can do that by sort of, well, visiting our website. You can become an asynchronous volunteer to get certified hours through professional nominations, graphic design, and social media promotion. We are also accepting team member applications if you want to take on a more active role in PHS to lead projects and initiatives to be up here with us. Once again, we are humbly asking that if you're financially able to donate, that you please consider sending a few dollars to us through to Venmo, via Venmo or PayPal. It costs a lot to keep our program up and running and free for all of you. So if you are someone who can afford to, or if you know someone else who can, please support those who cannot by donating to our organization so everyone can continue to get the education that they deserve. Otherwise, we simply ask you to spread the word about pre-health shadowing to reach as many students as possible. Now for the part that we've all been waiting for, earning a digital certification for the pre-health shadowing hours. Go onto our website and find our professional course pages. Next, you can take our quick 10-question multiple choice quiz based on the content from the session today. You will have 30 minutes per attempt to earn 70% or higher to get your certificate. We know that sometimes technology can be difficult, and for this reason, we do allow 2 attempts to take the assessment. But if you run into any difficulties, please contact us to ensure that our website does not please contact us. Again, to ensure that our website does not crash from a high influx of students, we recommend waiting 30 minutes to an hour after the session to take the quiz, after which it will be open indefinitely. Once you have passed, you can click the Finish Course button at the bottom of the professionals page and download your certificate, verifying your virtual shadowing hours. If you did miss a part of the session today, we do sort of have the recordings posted on our YouTube page. It will come up in 24 hours or so, so you can look at that. We also have recordings of our previous virtual shadowing sessions, so you can do those, and you can again get virtual shadowing hours for those recordings too. Be sure to follow us on social media and subscribe to our email list for updates. And you can also be sure to catch our sessions every weekday. We are also, thank you for joining us today. Please stick around if you have any questions. Me and the other team members will be happy to answer them. This shadowing session is officially over, and I invite you all to log off. Have a wonderful day.